Hello, everybody. Welcome back to another wonderful biology lecture. Today, we're going to be looking at things mutation and selection can do, because there are some things that it definitely can do. So, today's big questions are: what can evolution do? What do we know for a fact, beyond a shadow of a doubt, that it's capable of? And then, what are some commonly misrepresented evidences for evolution? Those are the two big questions for today. I think they're pretty good, and we're going to look at it. The new vocabulary for today is LTEE, or long-term Escherichia coli experiment, or E. coli experiment, and molecular homology. So good stuff. Homology, hopefully you remember from a previous lesson on evolution, our last unit, in the context of biology, refers to the similarity of structures or sequences of DNA, RNA, etc., in species that are derived from a common ancestor. That would be a good definition of homology. So looking at evolution, I'm going to go ahead and jump right into it. The first ability that it has is easily to cause changes to existing organisms. Okay, so if you have a existing species, say a wolf, for example, we know for a fact that through selective breeding, a more directed version of natural selection, you might call it selective breeding, that you can go from a ancestral wolf some 200,000 years ago to the specialized breeds of dogs that we have today. Evolution can theoretically do this, and we know it can because we have selectively bred and it was possible. So this is well within the ability of evolution to do. And depending on the definition of species, a wolf and, say, a poodle would be two different species of dogs, even though a wolf and a poodle could theoretically interbreed. Now, maybe a Chihuahua and a wolf couldn't interbreed, and the wolf might actually wind up eating the Chihuahua. But theoretically, if you took the sperm and the egg of a wolf and a Chihuahua, it would produce a viable wolf-Chihuahua hybrid organism. So in terms of reproductive viability, they're the same species. So changes within a species are definitely within the ability of mutation and selection to do, because we have, through human history, selectively bred dogs, wolves, to become domesticated dogs. In fact, there was a very interesting experiment done in Russia with Arctic foxes that you can look up and find out more about, where they selectively bred Arctic foxes for friendliness with humans and were able to domesticate them in a much, much shorter time than happened with wolves to dogs. They were able to create domesticated foxes within about 40 years, only a couple dozen generations. So very, very quickly indeed. And that's within the ability or the possibility of evolution to definitely be able to do. Now, the second thing that evolution can do, without a doubt, is changes to existing enzymes. Now, we talked about enzymes in our chapter on biochemistry and how they catalyze chemical reactions. Well, it turns out that there is a substance that humans have made called nylon. Okay, this is completely synthetic. It does not exist anywhere in nature. And bacteria have developed the ability, by altering an enzyme, to digest nylon. Okay, and of course the question is how many mutations were required. Bacteria have the ability to digest many hydrocarbons of many different types, so it may have required only a very minor alteration to the active site for a enzyme that catalyzes a reaction with a different hydrocarbon, because nylon is a hydrocarbon, synthetic hydrocarbon. But evolution can do this. We've had bacteria evolve this ability. It's a single protein enzyme change, but apparently This is within the capability of random mutation and natural selection and whatever adaptive programming exists within a bacterial cell to be able to do. Okay. Ability 3 would be new single protein functions. And a good example of this comes from the long-term E. coli experiment, Escherichia E. coli experiment, where they have been breeding E. coli for something like, over 80,000 generations. This is the equivalent of, 750,000 to about 1 million years of human evolution. Very long period of time, and its population sizes are in the billions because we're talking about bacteria. And what they've found is that after about 50,000 generations, sorry, it's hidden by the picture here. I need to adjust that. Maybe I will in the future and re-record this. After about 50,000 generations, they found that the E. coli could digest a substance known as citrate in the presence of oxygen. Okay, now what's interesting about this is that that would be considered for E. coli about a species level change. Because generally speaking, normal E. coli, or the E. coli they started with, can only digest citrate in the absence of oxygen. But after about 50,000 generations, they found that a specific breed or strain of the E. coli had developed the ability to digest citrate in the presence of oxygen. That means that some protein was altered to deal with free oxygen and thereby allowed the citrate digestion to occur in the presence of oxygen, where normally it would not. Okay, that's normally not something E. coli can do. And that's really interesting. But again, this falls under the same kind of interesting as the previous ability, single enzyme change function. This is probably due, or most likely due, to a single protein function where an oxygen transporter is being used to remove oxygen from the pathway, allowing the digestion of citrate to occur with the machinery that already exists in the cell that can digest this substance in the absence of oxygen. So evolution can definitely do this. Single protein functional change, single enzyme change. Okay, and this is just in the presence of oxygen. Again, this is the citrate molecule that's being digested. In the presence of oxygen, it normally can't be, but after about 50,000 generations, it was able to digest this. This happened along the way. Some other interesting things have also occurred. Yeah, I recommend you go to the website for the LTEE. There's a lot of very fun stuff to read in terms of what evolution can definitely do. But this stands out as the one that has the highest likelihood of being due to some kind of change to the actual program of the cell, as opposed to the degradation of the program of the cell. Okay, which mutations tend to be degenerative. They tend to destroy things. But in this particular case, it might have actually been a mutation that was in the affirmative. And the experiment has run now past 80,000 at the time of writing this slide. Forever ago, it was about 50,000. The citrate development came about 20,000 generations in. So very interesting. But something to keep in mind is that the generational cycle for a E. coli is about an hour. You can get an entire generation. It has population sizes in the billions, which humans, for example, have only gotten to in the last 100 years. And it doesn't have the mutation controls that eukaryotic cells have. Remember, prokaryotes like E. coli have relatively simple DNA loops, and they don't have a lot of mutation repair and correction mechanisms, whereas eukaryotes have a lot of mechanisms that will actually cause cell death if mutations are detected to any really significant degree. So huge population sizes, very short generational times, and mutation rates that are much higher than would be in a higher order organisms population, and it took 20,000 generations for this to occur. So very interesting, but honestly a bit lacking in terms of the types of advancement that you want to be able to see to, say, go from a common ancestor with primates to humans, right? You need some much more significant types of changes and multiple protein integrated type changes so that whole new functional systems, say the prefrontal cortex, can be both enlarged and developed in a way that allows for its functionality to enable the ability to create projections, to run scenarios that haven't happened yet, right? To estimate or to try to imagine, I guess, the ability to imagine fictional scenarios in which, say, you wind up dying because you're playing out the scenario in your head, right? Instead of actually doing it. And that's what the prefrontal cortex is expert at doing, is processing potentialities into allowing us to decide what is a good choice to make. And you're going to have not just the structure of the prefrontal cortex, but the programming underneath that allows it to do what it does in humans, which makes us so different from basically every other animal on Earth. Whales, for example, have brains that are much larger than ours, but they don't seem to have the foresight, the ability to project possibilities into the future in a fictitious way, in our imaginations, visualize things that haven't actually happened, that are gifted to us because of our relatively large prefrontal cortex. So, now 20,000 generations is equivalent to roughly 300,000 years of human evolution, assuming that the generational cycle for a human is 15 years. And this is give or take, right? Give or take 3 years. Humans probably want to be closer to 17 or 18 in terms of bearing children in a way that's not going to result in complications during pregnancy. Younger than 15, the more distant you are from 15 in terms of getting younger, the more birth complications you're going to have, because the female's hips haven't widened sufficiently and her body in general isn't going to be prepared to have children. At about age 15, 16, the body can theoretically have children and maybe not die from it in the absence of modern medicine. Right? Modern medicine has increased the survivorship of young pregnancies dramatically because of things like C-sections, where they can literally cut the mother open, take the baby out, and sew the mother back up, and her not die from a horrible infection, which prior to about 100 years would have been the case. So for a female human to be able to give birth to a healthy child and survive the birth, 15, 16 years is probably at about the minimum rate or minimum age you're looking at. But that notwithstanding, you can estimate around 300,000 years of human evolution, and what you get is the ability to digest citrate in the presence of oxygen. Really, you need more. And if evolution is going to be considered capable of doing all of the things that science says that it can do, you want more change in a shorter period of time than 20,000 generations, especially when you're trying to look at it in higher organisms like us, like mammals, or amphibians, reptiles, birds, fish, right? Bacteria can have huge population sizes, again, in the billions. Most other organisms only have population sizes in the hundreds of thousands, maybe millions. And humans, our population size has been probably under a billion for the majority of the entirety of human history. So very low population sizes and not a lot of room for excess mutations to accumulate, plus all of the machinery in our cells that resist mutation, working against the process of mutation, writing some kind of new beautiful program for, say, our prefrontal cortex. So Can evolution change things? In 20,000 generations, yes, it can definitely change one protein to deal with oxygen. Can it write an integrated system that gives rise to something like the prefrontal cortex in 300,000 years? That's a very different question, right? That's a very different issue that we're talking about. So the experiment continued to run. Its most recent published paper that I'm aware of, and again, I haven't looked at this in the better part of a decade, so it may have gone, well, I know for a fact it's gone to 80,000 generations, but the last published paper was at 50,000 generations that I'm aware of. And that's roughly 750,000 years of human evolution, again at 15-year generational times. And with population sizes that dwarf the entire history of human populations. So you want to see a lot more, to be honest. This is the very best that evolution can do, and it's really interesting. I don't want to make it sound insignificant. The ability of a single protein to get rid of oxygen so that citrate can be digested, happening by chance after 50,000 generations, that's really interesting, okay? And it definitely is pro-evolution can do things, right? It definitely supports the idea. But it's also interesting in the fact that it's only a single protein that causes the change. The rest of the machinery is there, and you don't see more change, right? You are seeing change, you are getting something, but it's not more. It's a little concerning in terms of generational times in higher organisms with populations that are far smaller and much more resistant to mutations as a whole. So keep in mind that E. coli could already digest citrate in the absence of oxygen. So this is not going to be a situation where a multi-protein system was developed, right? An integrated system didn't come into existence by these mutations to digest citrate in the presence of oxygen. It would have been far more compelling and interesting if the E. coli lacked the ability to digest citrate under any conditions. And then you have all of the protein machinery required to digest citrate and the protein machinery to get rid of oxygen happening or developing, evolving together simultaneously. You know, a multiple protein integrated system would be much more compelling and is the kind of thing that I'm looking for to justify the broader claims of evolutionary theory that say all life came from a single cell in the extreme distant past. All right, ability number 4. I'm sorry, I'm belaboring some of these topics and maybe talking a bit too much. Increased fitness due to functional loss. Okay, this has been well documented in a host of different situations. The first one we've actually talked about before, and it's sickle cell anemia. You have this parasite, the malarial parasite, that can attach to a normal red blood cell, a healthy red blood cell. Can have this parasite attached to it. The parasite drills into the red blood cell, eats it, multiplies, and kills it and spreads. And usually, without some kind of treatment, is going to be somewhat lethal to a human because it's a very nasty, it's killing your red blood cells, right? That's the oxygen transporting cell. Without oxygen in your blood, your cells essentially suffocate and you die. Now, sickle cell anemia is not a benefit. It is a genetic defect that can cause blockages like the one shown in this picture here in blood vessels, and can cause things like strokes, can cause things like heart attacks. It is not a healthy condition, but it does give you partial immunity to the malarial parasite because malaria cannot attach to a single sickle cell. It can attach to healthy red blood cells, but not sickle cells. Okay, and so someone who is homozygous for sickle cell anemia and is not producing any healthy red blood cells, they actually tend to die pretty early on in life because they get blood clots in a sensitive area like a capillary in the brain. Okay, and the brain, that part of the brain dies. It controlled, say, some essential motor function like breathing, and adios muchachos, you're gone. That's that's the end of your story. Now, we know that heterozygotes, those who are producing both healthy red blood cells and sickle cells, can survive, get over malaria pretty quick because the malaria can only spread at a very low rate due to the sickle cells, which then get carried on to the spleen and digested and killed. Okay, so this is a loss of function. It's a destruction of a healthy gene. The hemoglobin molecule, the gene responsible for the hemoglobin molecule, is being broken, and you get a positive benefit from it. That's common. This has been shown in corn. Another example is in antibiotic resistance in some types of bacteria. And the general idea here is you have your bacteria, you add antibiotics to it, and all but maybe one or two of those bacteria die because the bacteria, or the part of the bacteria that the antibiotics are attacking, are looking at, is broken. And so that bacteria survives because the antibiotic doesn't do anything to that particular bacterium. It thrives in the presence of the antibiotic. But if you take that bacteria and you reintroduce it to its host population that doesn't have the defect, in the absence of the bacteria, it gets out-competed. Okay, so it's a defect that allows it to survive a harsh environment, but in competition with the non-defective version of the bacteria is almost immediately out-competed. It doesn't reproduce as fast, and the healthy bacteria take over. Okay, so again, you get a benefit of resistance to a certain type of antibiotic, which will attack, say, a specific protein that's not being made in some bacterium. But that bacterium is not as fit as the regular bacteria in the absence of the antibiotic. Interesting stuff. So from the University of Brussels, a microbiologist, it says that throughout 150 years of the science of bacteriology, that's the study of bacteria, there is no evidence that one species of bacteria has changed into another. This is Alan H. Linton who said this, and it's from a quote in an April edition of the journal 2001. This may have changed since then. It's now 2025, and life keeps moving on. But we've been studying bacteria for a long time. We've been doing induced mutation experiments where we are artificially upping the rate of mutation for bacteria, and they have population sizes, again, that are vastly larger than maybe all mammals that have ever lived on Earth, depending on what type of study you're looking at. And you're not seeing the development of new multi-protein functionality, right? So 3 or more proteins that weren't working together before suddenly coming together because of an alteration of programming, the development of a gene regulatory network to make them work together, to do something brand new and novel, okay? And what you tend to see in nature is that instead of developing something new, like a special type of antibody to kill the malarial parasite, which humans have had thousands of years to evolve, we've been exposed to the malarial parasite for a very long time. Instead of developing something new, the way the human body has dealt with it is by allowing a defect to propagate in the population, sickle cell anemia. So I'm going to stop here, and we're going to look at some misconceptions in the next lecture This was a bit long. I apologize for that. So thank you so much for staying with me, and I will talk to you later.